All right, welcome. Welcome back, longtime viewers and listeners. Like and subscribe for more exciting content from Biology 112. Today we're going to look at Chapter 22, Continued Speciation, or Part B. So in this video, we're going to wrap up the chapter, and we're going to focus on those last two major topics that I introduced at the beginning of the previous video. The last two topics are, we're going to look at how the geographical distributions of organisms influence speciation, and then we're going to look briefly at how different genetic mechanisms produce new species. All right, so let's look at different ways that new species can form by focusing on how separated the groups are. So the first example, or the first term, is allopatric speciation. So allopatric speciation is speciation that takes place when a physical barrier divides a large population, or when a small population becomes separated from the larger population. So to translate that, allopatric speciation happens because something in the geography or something in the environment separates two subpopulations, and over time those two subpopulations become more different and less genetically similar and become separate species. All right, so if you look at the picture at the bottom of this slide, you kind of see you can see how allopatric speciation can happen. This is a pretty good explanation, I think. So let's begin with one population. So each of these black dots represents an individual in this population of whatever animal you want it to be. And then something happens geologically, well, whatever, both, that separates this one population into two. So in this case, there's a new river that's formed for whatever reason. So this river over time is going to become a lake or a larger body of water. And this is a geographical barrier. So these individuals are going to have trouble getting over here to mate with these individuals and vice versa. All right, and then here, over time, you can see that because individuals from the two populations aren't breeding, that's going to cut out gene flow. Right? And if a mutation pops up in this population, it's going to stay here. And if a mutation pops up in this population, it's going to stay here. They're going to have different selective pressures. Right? The populations will be of different sizes. So all those mechanisms of evolution that we learned about earlier are going to be in play, and they're going to be affecting these two populations differently. They're going to affect the allele frequencies of this population in a different way than they're going to affect the allele frequencies of this population. So over time, the two populations become less genetically unique or similar, less genetically similar. And then if something happens, let's say this reverses and this lake drains away and the two populations come into contact again, they might have enough differences, whether they're behavioral or ecological or genetic or whatever. They're going to have enough differences because of their time in reproductive isolation that the two populations are no longer genetically compatible, or they're no longer behaviorally compatible, or whatever. The point is they don't mate anymore, and so they're now two distinct populations. So this is an example of allopatric speciation, where geography splits one population into two, and the result is two distinct species. The bottom picture is some fish, and this example happened in real life. So here we are zoomed in on the map. Here's South America, here's Central America. And when sea levels were higher in the distant past, the Atlantic and the Pacific Oceans were connected, so there was no land bridge here in Panama. And then as sea levels receded, went down, then the two oceans, their levels receded and exposed dry land connecting Central America and South America. And this basically split the Atlantic from the Pacific in this region. And so initially there was a species of fish. This is a wrasse. I think I'm saying that correctly. Race? I don't know. How do you say that? And initially it was one population, one species, just like up here. And when the land bridge formed, some of the population was trapped in the Atlantic or the Caribbean, and the rest of the population was trapped here in the Pacific. So over time, just like here, this population bred, and certain mutations happened theoretically, and basically the alleles shifted in a particular direction that caused them to become blue. And over here, this population, those same things happened, and the alleles shifted, and they became red or rainbow. And so now we have two distinct species of these fish, one in the Atlantic and one in the Pacific. And if they're brought into contact, they will no longer mate and reproduce, and so there are two species. So this is an example of allopatric speciation. We can also see allopatric speciation play out in a species cluster. So a species cluster is a group of closely related species recently descended from a common ancestor. And so to illustrate a species cluster, we can return to the example of Darwin's finches in the Galapagos, or any species in the Galapagos or any other island chain. So this image is from your book. So we've got members of a species on a mainland, and then for whatever reason, a few of them land on two of these islands. So maybe a storm blew them out there, or they migrated, or whatever. And then, sorry, drinking some coffee. And then over time, these individuals, because they're allopatrically separated, they're on different islands, they become two different species. And then species B, in this case, moved to island C and D. And then we got some founder effect going on. And over time, they became distinct species. And then, so in this example, we've got 4 islands, each with their own unique species. And then over time, because they've been separated for so long, these species become distinct. And so now if they're reintroduced to an ancestral species or a cousin species, there's enough genetic or behavioral or ecological or temporal or mechanical differences that would prevent them from mating and having viable offspring. So these species here are a species cluster, basically a bunch of species that have evolved from a recent common ancestor. By the way, we see this happening in this situation frequently. So in other words, a lot of times our species clusters are going to show up on these island in these island systems where you got some allopatric speciation going on, some geographical separation. Excuse me. All right, sometimes you can test whether allopatric speciation has occurred. All right, and what I mean by that, let me go back to this one. If these populations are separated, right, you can get some divergences. And then when they come into contact, if, let's say it's a fairly recent separation or a short-term separation, when they come back into contact, then they might still be mating with each other, and the alleles can still be flowing, can start flowing again between the populations. And so maybe there hasn't been enough time or genetic differences to cause 2 new populations. So that's where secondary contact and species fusion come into play. Secondary contact is simply contact after a period of geographic isolation. Going back one more time, sorry. This would be secondary contact. When these 2 populations are split and they come back together, that's secondary contact. Why do we care? Well, we care because this is a test of whether or not those 2 populations have diverged enough to make them reproductively isolated. So again, if there hasn't been enough divergence or there hasn't been enough time, then after secondary contact, then those 2 populations are going to return to being one species, to being one gene pool, right? And so if that happens, then the 2 populations will interbreed and turn back into one species, one population, and that's called species fusion. All right, so let's see. So here we've got allopatric speciation that's that split. Sorry, I got a kid crying in the background. Sure, I'm sure she's okay. Um, Man, sorry, really distracting. Have a kid screaming. My wife's home though, so it's probably fine. So we got two groups. They're split geographically, and then over time they come back together. So here's secondary contact. That's bringing the two populations back into contact. In this example, we have hybrids that are forming. So maybe there's not enough prezygotic isolating mechanisms in play, and so some hybrids are formed. But let's say those hybrids are invariable, and so you get hybrids that are being born, and the hybrids are maybe they can't develop or they're less fit, and so the hybrids get selected against. So in this case, these hybrids die, and it effectively creates a barrier between these two groups. And so in this example, this secondary contact did not lead to species fusion because these hybrids were selected against, and so the greens remain green and the blues remain blue, and we have two new species that are the result initially of geographic isolation. So here we have allopatric speciation, right? That splits the population into two because of geography. And then we get some post-zygotic isolating mechanisms following secondary contact. And so that leads to these two populations remaining distinct and ultimately becoming two species. I skipped a couple things in the book, but you don't probably have your book. It's probably still in your dorm room, so you don't know the difference. All right, the next, and sometimes what can happen is you can form these hybrid zones. So going off that example we just discussed, here we've got these oriole birds, all right, and so here is the Bullock's oriole, and this is the Baltimore oriole. So you can see the Baltimore oriole lives basically east of the Rocky Mountains, and Bullock's oriole lives in the Rocky Mountains and west. And where their ranges overlap, you can form these hybrids. And so these are called hybrid zones. And so hybrid zones are basically just places where as two species interbreed, they produce viable and fertile offspring. Right? So these hybrids are variable, they can survive, they can reproduce. And so these hybrid zones can persist for a long period of time. And these hybrid zones kind of serve as an intermediary where genes can flow between these two populations or between these two species. But anyway, okay. So let's talk about sympatric speciation. In sympatric speciation, we get reproductive isolation between distinct subgroups that arise within one population. All right, that definition's terrible. Let me go to the graphic and try to explain it. So in the first one, I pulled this off Google. In the top example here, top picture, you can see allopatric speciation. So one population that here a canyon is formed, and it's now become two. And the two subpopulations on each side of the canyon over time, because of reproductive isolation, they become two distinct species. Right? Classic example of allopatric speciation. Here we have sympatric speciation. We've got one population, and within that population we get a new subpopulation, eventually becoming a new subspecies or new species that's forming. So in sympatric speciation, you have speciation, but the two populations are not separated geographically. They might overlap and be in the same location. All right. So a good example of sympatric speciation is insects that feed on, let's say, one plant species. So imagine an insect, and in the picture, you can see an apple maggot. That's gross, but they're basically flies, and they lay their eggs in. I got a kid screaming now. Whew, she's angry. Can you hear that? I don't know why I ask questions. I can't hear your answer. Oh my word. It's my 4-year-old. So this is a fly that reproduces on a hawthorn leaf, which is confusing because it's called an apple maggot, but whatever. All right, so a hawthorn's a type of plant. You can see the leaf here. So this is the female fly and the male fly, and so they'll hang out on the hawthorn plant. They'll eat it. Maybe they'll eat the fruit. They'll lay eggs on the hawthorn plant. Their eggs hatch into larvae or maggots, and the larvae eat the hawthorn plant, develop into adults. So basically, they spend their whole life cycle on this plant. Imagine, if you will, that some of these flies fly to, they leave a hawthorn plant and they fly to a new species of plant, maybe that's growing next to the hawthorn. Let's say they fly to an apple tree. And those flies start doing this same life cycle on the apple tree. So over time, maybe that second group of flies will become adapted to the apple tree. And so their young will eat the apple leaves, and maybe the adults will feed on the apple fruit. And so this is an example of sympatric speciation. We now have two species of flies, one on the hawthorn and one on the apple. And they're not separated geographically. They could fly, those two members of those two populations could fly and interbreed if they want. But because they're living on different plants, and because over time they're becoming adapted to the different plant nutrients and compounds, the flies eventually become two species. So again, that's an example of sympatric speciation. Sometimes we'll talk about host races. So initially, when those flies left the hawthorn and landed on the apple tree, we would call them a host race. So in other words, it's like a race of flies initially. It's not a new population. It's a race that's adapted to a different host. So you have a host race that's adapted to hawthorns, and a host race that's adapted to apple trees. So this is the beginning step of sympatric speciation. So they're splitting, again, not geographically, but they're splitting based on their food source. And they begin as host races, and over time, they might become new species. I pulled this off the Google. This is showing allopatric, once again, ancestral species, barrier, so then we get a new species on one side of the barrier, and in this case the ancestral species remains the same on the other side of the barrier. In sympatric, you get your ancestral species, so maybe this is the flies feeding on the hawthorns, and then you got a new host race that is feeding on the apples, and then over time they become a separate species. So again, sympatric speciation is just speciation in the absence of a geographic barrier. All right, part 4, let's look at genetic mechanisms of speciation. Oh, there's an error in my, this heading should be the genetic mechanisms of speciation. I just noticed that, sorry. Let's talk about genetic mechanisms of that cause speciation, in some cases really rapid speciation. Okay, so the first term to wrap our minds around is polyploidy. So polyploidy is simply a genetic phenomenon where an individual has extra copies of chromosomes. So in this example, here's our normal karyotype. So the karyotype is simply the chromosomes that are represented in that species. And here you can see we've got triploidy. So instead of there being 2 homologous chromosomes, there's 3. So this would be the consequence of an error in meiosis. So the resulting organism is going to have 3 copies of each chromosome instead of 2. All right? So that's triploidy. It's got 3 copies. But it's an example of polyploidy. Polyploidy, again, is just they've got 1 or more extra sets of chromosomes. Okay, why do we care? We care because this individual, because it has extra chromosomes, it can become an entirely new species. In other words, it becomes, especially post-zygotically isolated from this species. And so over time, this will remain a species, the ancestral species. And then this polyploid individual will become a separate species, again, because it's genetically incompatible with this one. And so how will this one mate? Well, oftentimes in plants, it will self-fertilize. So this one will not be this one will not be compatible with this one, so it'll self-fertilize. And so its offspring will have that extra set of chromosomes, and again, over time, it becomes a separate species. I'm not going to talk about trisomy and monosomy. I can't pronounce any of these, so just ignore those. But what's interesting is a lot of the fruits and vegetables that we eat, for whatever reason, are polyploidal. So in the breeding processes or in the domestication of wild plants to domesticated varieties, polyploidy often leads to larger fruit, and polyploidy is pretty frequent in most of the domesticated varieties of fruits and vegetables. You'll see that with wheat in just a minute. So speciation can occur fairly quickly, right? So hopefully this paragraph makes sense. When two populations are separated geographically, right, then those populations are going to accumulate genetic differences through the mechanisms of natural selection that we talked about in the last chapter. And so, but just know that sometimes it doesn't take a lot of genetic differences to accumulate. Sometimes it can just be a single mutation or some just shifts in allele frequencies that can cause enough differences that create new species. So the example of that is these ducks. So we've got mallard ducks and pintail ducks. So they're both members of the same genus, but they're different species. And so these are closely related, but they're separate species. And the way that it's thought that they came about is that there were just different sexual selection pressures. So in the mallard ducks, there was sexual selection for the males to have brightly colored green heads, and the pintail, it was brown. And so that's pretty minor genetic differences between the two, but sexual selection has shaped the males in a way that's going to cause these two populations initially to be isolated, and then over time enough genes and mutations and so on build up that sexual selection is enough to drive these two populations in a direction where it leads to two new species. Another example from the book is in this species of snail, and I won't try to pronounce its species name. It's crazy. What happens is there's a single mutation that occurred in the population, and this single mutation caused the shell of the snail to develop in the opposite direction. In other words, in the ancestral population, the original population, the shells developed in a clockwise spiral. But the single mutation causes those snails to grow a shell that grows in a counterclockwise spiral. And that might not seem like a big deal, but the difference in the direction of the shape or the growth pattern of the shell prevents them from mating. And so it's basically mechanical isolation. The clockwise snails can't mate with the counterclockwise snails. So this is an example where a single mutation created mechanical isolation. And so you get speciation that happens not over a long period of time, but basically it pops up really quickly in a generation. So that would that be allopatric or sympatric speciation in the snails? Notice how I gave you time to think about it and answer. The answer is sympatric, right? Because the counterclockwise and the clockwise snails aren't separated by a geographical barrier. They're going to be side by side, so it would be an example of rapid sympatric speciation. So back to polyploidy. There's two subcategories that we need to talk about: autopolyploidy and allopolyploidy. So actually, let me go to the next slide. In the top picture here, top image, this is how autoploidy works. So well, let me define autopolyploidy. In in autopolyploidy, a diploid individual will produce a polyploid offspring. And what happens is that occurs because of a mistake in meiosis. So we've got an individual that produces gametes that have extra chromosomes, so they're polyploid. And we would call these gametes, let me go back, we would call these gametes unreduced gametes. So look at this example. So here's our original cell, so it's diploid. So the diploid value is 6. Excuse me. I'm okay, it's allergies. And then there's an error in meiosis that leads to gametes that are unreduced. So if you remember from meiosis, we're supposed to go from the diploid number, in this case it would be 6, to creating gametes that have a haploid number, which in this case would be 3. But there was an error, and our gametes still have 6 chromosomes each. Uh-oh, that's twice as many. So in this case, the individual might be infertile or unable to reproduce with other individuals that have the correct haploid number. So this, let's say this is a plant, so then it undergoes self-fertilization. So this unreduced gamete fertilizes this unreduced gamete. Basically, this plant mates with itself, and the end result is a zygote that has 12 chromosomes. So in one generation, we've gone from a diploid number of 6 to 12. So if this 12 chromosome zygote can develop and become a new plant, this is an example of how autopolyploidy can create a new species just in one individual or in one generation, right? Pretty rapid. Another example is allopolyploidy. So allopolyploidy, which other than being a difficult word to pronounce, is when two closely related species hybridize and then form polyploid organisms. So let's work through the example down here. We've got species A and species B. You can see they both have a diploid number of 6, but they're distinct species for other reasons, all right? So mutations or whatever. Through meiosis, we don't have an error. Through meiosis, we end up getting a gamete as we would expect with a haploid number of 3 and a haploid number of 3. And then the pollen or whatever, the gamete from this species fertilizes the gamete from this species, leading to a hybrid with the correct diploid number once again. All right? But because these chromosomes are from 2 different species, it causes problems in mitosis. So it ends up creating, as this zygote undergoes mitosis, it ends up making cells that have 12 chromosomes in them. So it picks up additional chromosomes in the hybrid. Then when that hybrid produces gametes, its gametes now have 6 chromosomes each, right, which is greater than the original number of 3. And then if it was to self-fertilize, then you would get an offspring that has 12 chromosomes instead of 6. So you can see that in allopolyploidy, you can generate a new species, and it would be a new species because it has a different number of chromosomes, but it does that through the production of a viable hybrid as an intermediate. Right? The end result is kind of the same in autopolyploidy and allopolyploidy. You end up getting a new species that's polyploid, but just the mechanism by which it's happening. One, it's just kind of a mutation or an error that's happening in one species in autopolyploidy. Auto means self. And then in allopolyploidy, you get a new species forming with a hybrid that, as a result of a hybrid forming from 2 species successfully hybridizing. Okay, so wheat is an example of a polyploid plant. So it's thought that the original ancestor of wheat gave rise to two diploid lineages. All right, so here's our, we don't know what the original species was, but it gave rise to a species that here it's represented by having two A's representing the alleles, and this population has two alleles represented by the letter B. In both cases, they have the same number of chromosomes, so a diploid number of 14, but over time, these bred and created hybrids, so AABB, right? Or over here, we got a descendant that produced a new diploid lineage with genome DD, right? And then that gave rise, then these crossbred, and it gave rise to one that was AABBDD, so a polyploid descendant. And the result was, this is basically a new species of wheat, right? And so all of the cultivated wheat that we have today, it's thought to have arisen from this process. So basically, a wild type wheat ancestor that produced polyploid offspring through subsequent generations, and over a long period of time, those hybrids were able to be bred, perhaps artificially by humans, to create the domesticated variety of wheat that's polyploid. It's got 42 chromosomes, whereas the wild ancestor probably only had 14. So wheat is an example, is one example, but again, many of our domesticated plants that we use in agriculture are the result of polyploid fusions. So hopefully you're catching on that these chromosome rearrangements can lead to speciation, and in some cases can lead to rapid speciation. So to illustrate that point, let's just step back and look at or compare the karyotypes or the chromosomes of a mouse and a human. So here we're using the human chromosomes kind of as our base. So you can see here's all the autosomes numbered, and here's the sex chromosomes. In this case, it's an X and a Y. And so all of the genes that we find on chromosome 1 are coded, what color is that? Dark green. All the genes that make up chromosome number 2 are coded regular green, and so on and so forth. When you look at a mouse, right, you can see a couple of things. One, it has 19 autosomes, or we have 22. So mice are different from humans. Why? Well, one reason is they have different numbers of chromosomes. So this would confirm that humans and mice are separate species, so we're not genetically compatible because of the different number of chromosomes. But when we look at the chromosomes, we see that mouse chromosomes are kind of a mashup of human chromosomes. So what we'll call human mouse chromosome number 1, it has sections from human chromosome 6 and human chromosome 2 and human chromosome 18 and human chromosome 1. So in other words, it's not just the number of chromosomes that lead to the production of a mouse versus that of a human, two distinct species, but it's also how the genes are arranged on those chromosomes. So simply by mixing up the locations of genes, you can create new species. Right? And so humans and mice inherit a lot of the same genes, but they got mixed up over time, and so they are on different chromosomes and in different orders, and the differences between mice and humans is in part because just the arrangement of the genes is different. To get more specific, let's look at humans versus chimps, gorillas, and orangutans. And by the way, you see this banding pattern. So what you can do is you can just stain the chromosomes, and different genes will kind of stain dark versus light. And so that's what they've done to kind of, let me go back, no. And that's what they've done here. They've taken chromosome 2 from a human, and they've stained it so you can see the banding pattern. It's just a way to visually compare chromosomes to each other among different species or within a species. All right, so this is human chromosome number 2. So you can see the banding pattern, and you can see there's a centromere here. All right, and hopefully you remember what a centromere is. And if you look at a chimpanzee, it has a chromosome that has the same banding patterns, or very similar banding patterns, as this section of human chromosome 2. And then they have a second chromosome that lines up with this half of human chromosome 2. So Why are humans and chimpanzees are separate species, but were considered fairly closely related evolutionarily. And one difference, or one thing that happened in the lineage, the common ancestor of humans and chimpanzees, is that there was a chromosome rearrangement. And so somewhere along the line, these two chimp chromosomes fused together in a particular population, perhaps, and gave rise to, instead of having these two separate chromosomes, it gave rise to human chromosome number 2. So then as that population would interbreed, individuals that had 2 chromosomes eventually evolved into chimpanzees, and individuals where the 2 chromosomes fused into 1, they were more compatible, and so they eventually evolved into humans. So the speciation of humans and chimps is in part because of this fusion. All right, and then if we look at gorillas and orangutans, you can see that they also have 2 chromosomes. But what's interesting here is, so this chromosome on a gorilla is similar to this chromosome on a chimp. But on this chromosome, this section is similar to this section of the chromosome on the chimp. But this section in the yellow box is flipped. You see that? This part on the left on the chimp matches this part on the right on the gorilla. And it's the same as the orangutan. So somewhere in the common ancestry where it split to create gorillas and where it split off to create the common ancestor of humans and chimps, there was an inversion that caused this part of this chromosome to flip. So chimps and humans both inherited this flipped version from a common ancestor that was a separate population or a separate species from the individuals that gave rise to the gorillas and orangutans. You can see it again here. This is the same data. So gorillas and orangutans inherited chromosomes that look like this and this, whereas there was an inversion event that gave rise to the common ancestor of humans and chimps. So again, the inversion event was this got flipped to look like this. And then somewhere in the human lineage, there was a fusion where these 2 chromosomes fused into 1. So again, we can use these, couple things. We can use these chromosome rearrangements to track evolutionary relatedness, right? We use it as an explanation of why humans and chimps are so considered, closely related. And it's also an explanation for why we get these different species. So the ancestor of chimps and humans split off from gorillas because of the inversion, and then humans and chimps split off in part because of the fusion event. And by the way, talking about banding patterns, we looked at humans versus mice, and the image on the left is humans versus chimpanzees. So this is a human chromosome, and this is chimp chromosome. So just based on the banding patterns, you can see they're really similar. So if you work your way through this karyotype, you see that humans and chimps aren't just similar in the way that our nucleotide bases are arranged, the A, C's, T's, and G's, but we're also very similar in our banding pattern. It's not just that the genes are similar, it's also that they're found in more or less the same places on the chromosomes. So this is also used as evidence of human and chimpanzee relatedness. And then, of course, going back to what we were just talking about, here's human chromosome 2, and you can see that that this chromosome is the result of the fusion of this chimp chromosome with this one, right? Just fuse them together, and you're going to create human chromosome 1. So these chromosome rearrangements lead to speciation. All right, and that sometimes that can be really quick. All right, so hopefully polyploidy and chromosome rearrangements give you a better idea of how speciation can happen pretty quickly. It can lead to gametic isolation or postzygotic isolation. And again, all of the things we talked about in this chapter are different ways that new species can form. All right, like and subscribe. And if you got any questions, see me at the Zoom chat or post them in the discussion board.